human body is made of cells, trillions of cells, and those cells are specialized and they have particular jobs to do. So like nerve cells transmit impulses, muscle cells contract. But one thing that all cells have in common in your body is that they have a plasma membrane, and that plasma membrane will allow some materials to enter the cell. It'll allow some materials to exit the cell. It'll keep some materials in the cell. It'll keep some materials outside the cell. But in order to act with this environment, it's got to be able to have a membrane that allows some things in, but not everything, and allows some things out, but not everything. Those processes of things entering and leaving the cell, we call those osmosis and diffusion. Diffusion has to do with the movement of molecules and particles and atoms and ions in just a random kind of way. Osmosis has to do with strictly the movement of water. And it always tends to move from a greater concentration to a lesser one. To illustrate this, like a big catch from a fish. Diffusion and osmosis are just physical processes that act inside the cells of organisms. Diffusion means simply the movement from an area of high concentration to an area of low concentration. Osmosis is a specialized form of diffusion which involves the diffusion of water. Active transport is different from diffusion because it requires energy to move molecules against the concentration gradient. So diffusion is the movement of particles from an area of concentration to concentration. So in the case of diffusion, it's always moving from an area of high concentration to an area of low concentration. The plasma membrane is important in playing a role with osmosis and diffusion because it allows things to get in and out of that membrane. But let's take a careful look at this cell membrane. Here's a few things to notice. Notice that the cell membrane consists of some carbohydrates. The carbohydrates can be connected to a protein. The name of that is going to be a glyco, that's the glycogen, that's for a carbohydrate, protein, glycoprotein. They're embedded in the cell membranes and they help the cells communicate, they help the cells be marked chemically so that other things can interact with them and materials can identify what kind of cell that is. For example, hormones. Here's a cholesterol embedded in the cell. So sometimes people say, oh, I have too much cholesterol, I got to get rid of it. And that is a problem because it can cause problems with your arteries and veins. But what if you had no cholesterol in your body? Well, without any cholesterol in your body, you would have trouble maintaining your cell membranes. They'd start falling apart. So we've got to have a little bit of cholesterol, just too much is unhealthy. A sugar carbohydrate attached to a fat molecule is called a glycolipid, because the lipid refers to the fat molecule. Notice that I have a layer of lipids here, and I have a layer of lipids here. So I have a lipid bilayer, 2 layers of lipids. There's my 1 layer on the top, 1 layer of lipids on the bottom. Notice I have some cytoskeleton on the inside of the cell, maintaining the integrity of that cell membrane. And notice that I have some channels or gates. So some proteins can make doorways or windows. We could think of it like entrances and exits in our cell membrane as well. One of the first things I think of when I think of a cell membrane is that lipid bilayer. 2 layers of lipids, phosphate heads, lipid tails. And I think of it like a double-stuffed Oreo cookie. Now, why does it have to be a double-stuffed Oreo cookie, not just a normal Oreo cookie? Well, it's because the lipid bilayer, there's 2 layers of fats. So the chocolate crunchy cookie would be like the phosphate heads, and then the 2 layers of white creamy goodness on the inside are like the lipid tails. Now, it is important that it's a lipid bilayer, because you've probably heard the old saying, oil and water don't mix. Well, oil is a lipid, and oil and water, lipids and water, they don't mix. They tend to separate. So because of this lipid bilayer, it keeps the water inside the cell separate from the water outside the cell. The water in the extracellular fluid, that's outside the cell, and the water in the cytoplasm, that's the inside the cell, are separated by that lipid bilayer. The other thing about cell membranes is that they are a fluid mosaic. Mosaic means that they're made of more than just one part, like a mosaic in art or a music mosaic. There's more than one kind of music. There's more than one piece in the mosaic for art. There's more than one part to the cell. There's proteins, there's cholesterol, there's fat. There's all kinds of parts to it. There's gates, there's channels, there's carbohydrates, and these glycoprotein configurations. So there's a mosaic. There's lots of different parts. But it's also fluid. That means none of those parts are in any fixed location. They're constantly moving around the membrane. So picture your room, the room that you're sitting in, or a room that you are familiar with. So picture that room. In that room, you have doors and windows and walls. We'll say that the walls and doors and windows make up the plasma membrane. The walls and doors would be things like the protein channels and gates. Now imagine that it were a fluid mosaic. So it's not just made of brick and wood, that would be one material, but because it's made of brick and wood and other things, it's a mosaic. If it were a fluid mosaic, those doors would have no fixed position. So at one moment, the door is right in front of you. A little while later, it might be migrating across the ceiling over to the other side of the room. You might never know where the door is, which could cause you a lot of injuries if you were bouncing around in the middle of the night. But for cells, that's how cells work. They are a fluid mosaic. The third major part about a cell membrane, or concept about a cell membrane, is that it's a plasma membrane and that it's semi-permeable. That means that some things can enter, but not everything. And that some things stay in, or can leave, but not everything. And it reminds me of a tea bag. Because when you put a tea bag into water, you expect the water to go in and out of the membrane freely. The tea leaves, on the other hand, you expect them to stay in the tea bag. The tea flavor, you expect to go in and out of the bag freely. So it's semi-permeable. Some things come in, but not everything. Some things go out, but not everything. Like a tea bag. So let's label some parts of the cell membrane. So here we have a carbohydrate. It's kind of hanging out here, carbohydrate. Here we have a carbohydrate attached to a protein. That's a glycoprotein. Here we have a protein gate that can allow special things to come in and out of the cell. Here we have some cholesterol embedded in the cell membrane, helping maintain the membrane integrity. Here we have the lipid bilayer, that's 2 layers of lipids. Those glycoproteins and carbohydrates on the cell, they act to allow cells to recognize, not just each other, but for other molecules to recognize that this is the right cell to go to. But there are other things that help cells recognize each other, and more than that, they help cells stick together. It's kind of like rebar in concrete. If you have concrete blocks, that's fine. You add some rebar in there, some metal beams in there, and that concrete will become really strong. Cells are similar. Cells that recognize each other will form these bonds so that they become a tissue. The thing that's holding those cells together is a protein called laminin, one of the cell adhesion molecules. That helps form tissue, and this is what it looks like. So you can think like one day you wake up and you get a text, and your text says from your boyfriend or girlfriend, I'm going to stop dating you, I don't like you, I never want to see you again. You're like, oh man, my girlfriend or boyfriend just broke up with me today, it's horrible. And you go downstairs to get your breakfast and you burn your toast and you're like, oh, this day is terrible. And you go to get in your car and your car doesn't start and everything, the wheels fall off and it's horribly like, oh, my life is falling apart. And just as you walk back inside, you hear a knock on the door, it's from a bill collector, and they're like, you forgot to pay this bill, and oh, you're like, my life is falling apart, it's falling apart. But when your life is falling apart, you can be thankful to remember that something like laminin is actually holding your life together. It's keeping you together. So no matter how bad things might seem in your life, something like laminin is holding it together. Here's another diagram of what laminin looks like, which is kind of inspiring when you think that here's a molecule that's holding you together. The cell membranes are semi-permeable and have lipid bilayers. So we might ask, what can diffuse through it? So we look at some of these things. What could go right through a lipid bilayer? At first you might think, well, water should be able to, right? But remember, water and oils don't mix. Large proteins are too big. Cookies, of course, are too big. But lipid steroids can slide right through because they're lipids too. So let's take a look at some of these concepts, osmosis and diffusion. So here I have a dropper full of, we'll say, food coloring, and here I have a beaker of water. So I'm going to add the food coloring to the water. Let's watch what happens. So I get a little drop. The drop starts off very concentrated, and it moves through the water. So make some observations about what happened there. Let's take a look at what happens when we put some food coloring into some water. So here I have some red food coloring. Notice that as the food coloring comes out of the bottle, it's a very concentrated drop. And then watch what happens as it interacts with the water. So see how it's gradually diffusing through the water. Now these are just random movements of particles. Now of course every time I shake it, it kind of speeds up the movement, but right now the particles are just moving randomly and are distributing themselves in kind of an even way. Let's take a look again. We put the drop of water into the food coloring. When we add that drop of water to the food coloring, we say that that drop of food coloring is concentrated, or we say it's concentrated. Then, once that food coloring hits the water, we say that it's become diluted. It's not nearly as concentrated as it used to be. We diluted the food coloring by putting it in so much water. Now we can figure out how concentrated something is, how dilute something is. Here's how we do it. We take the mass of the solute. The mass of the solute is the thing being dissolved. So let's say I took a lump of sugar and I dropped it into water and the sugar was dissolved in the water. The sugar is the solute. The solvent is the water, the thing doing the dissolving. In this case, the food coloring is the solute. The water is the solvent. It's the thing doing the dissolving. So I take the mass of the sugar, salt, food coloring. I take the volume of the solvent, the water, whatever it happens to be. And when I divide the solute by the solvent, that gives me the concentration. So if I take 5 grams divided by 100 milliliters of water, so I put 5 grams of food coloring into 100 milliliters of water, then I get this small amount of a concentration. If I take 10 grams, then I've increased my concentration. I've gone from 5 to 10. If I take 20 grams, then I've increased my concentration. I've gone from 10 to 20. So if I put more solute in, then my concentration goes up. So if I put more salt in the water, I get saltier and saltier and saltier water. The concentration of salt goes up. If I add more water, then the concentration goes down. I've diluted the water. So when we look at a college student who has been partying and she takes some ecstasy, and one of the problems with ecstasy is it retains water in her body, making her blood pressure go up and her potassium, chlorine, sodium levels go down, we might ask, why would that happen to a person? Well, think about it. If her body's retaining water, the concentration of the water is going up and diluting the concentration of the things being dissolved, like sodium, chlorine, potassium. So those levels go way down. Let's take a look then at simple diffusion. So here, I have a beaker that has a high concentration of something on this side. Well, salt or sugar or food coloring, something on that side in a high concentration. There's a barrier between the two that doesn't allow the molecules to move to the area of low concentration. What happens when I move that barrier? Well, the molecules move from a high concentration to an area of low concentration. That's just simple diffusion, going from an area of high concentration to an area of low. So what happens if I have 3 grams of salt and I dissolve them in 10 milliliters of water, because milliliters are the same as centimeters cubed. So you got to do a little quick math, and hopefully find out the answer is A. Now there's lots of examples of diffusion in the human body, but let's just take this one. Here we have a high concentration of oxygen in the air. We inhale the air into our lungs. Now we have a high concentration of oxygen in our lungs. The oxygen diffuses across those thin membranes in our lungs to capillaries, where blood cells can pick up that oxygen. So the blood cells traveling the capillaries pick up that high concentration of oxygen. The capillaries move through your body, and again, they're going to areas of low concentration. So the oxygen is being dropped off. It's just diffusing in areas of low concentration throughout your body. So it's always going from an area of high concentration to an area of low concentration. Now, if you were talking about carbon dioxide, it would be going in the opposite direction, right? The high carbon dioxide buildup in the cells will be picked up by the blood in the capillaries, transported back to the lungs, where it will be dropped off because there's a low concentration of carbon dioxide in the lungs, and we breathe it out. So oxygen gets into our body by diffusion from the lungs. Now osmosis is the movement of water, and it's kind of weird to think about a little bit. But over here on this side, I have a high concentration of water. Why do I know that? I know that because there's more water in the solution than there is, let's say the red dots are sugar. We could use sugar or salt or anything, but let's just say sugar for the sake of sanity. Here I have a semi-permeable membrane, which is going to allow water to travel back and forth, but not the large sugar molecules. Here I have a low concentration of water. One way to look at it is kind of like this. This is my container on the one side. That's my container on the other. On this side, let's call this side A, we'll call this on side B, I have such a high concentration of sugar, I don't have much room for very much water. On side A, I have a low concentration of sugar, and since I don't have much sugar in there, I have lots of room for water. So when I look at A and B, I might ask which has a higher concentration of sugar, and you would say B. And if that membrane were removed, the sugar would move from B to A. But we're talking about the movement of water. So now I'm going to ask which has a higher concentration of water. And you would say A. There's way more water in A. So what's going to happen now in this membrane? Across this membrane, in this U-shaped tube, as the water moves from side A to side B, I get this strange looking distribution. The only thing that's happening is that the amount of water is evening out on both sides. So that some of the water from A is moving to B. Now that might seem kind of strange, but we're talking about the movement of water. Since the sugar can't move, but the water can. The water is going from an area of high concentration to an area of low concentration. So if you have half a glass of sugar water, and it has a concentration of 4.5 grams per milliliter, and you had half a glass of sugar water with a concentration that's much stronger, the new solution would be more concentrated. Sometimes for cells, this gets to be a problem when it takes a long time for things to diffuse out of the cell because it's just gotten too big. So cells are typically very small, so they can maintain a good surface area to volume ratio. So long as the surface area stays big and the volume stays small, the cell can interact with its environment, no problem. If the cell gets too big, then it's too far. It takes too long for things to diffuse. Imagine it like this. Let's say you're held hostage with a group of people in a building. And the people that are holding you allow you to carry a bucket from your room to the bathroom. And you can dump the bucket in the toilet. Let's say after a while, though, the bathroom stops working. So instead of being able to carry your bucket just down the hall to the bathroom, now you have to carry your bucket 2 miles away and back. So as you're walking down the street with your bucket, all the other prisoners that are stuck in the room have to use the bathroom. They can't go in the bucket because you have it and they're waiting for you to get back. Now if it takes you an hour to go and get back, it's uncomfortable, but maybe it's bearable. What if you had to walk 10 miles away? Now you have people who might be, instead of waiting for you to get back with the bucket, they might be taking a leak in the corner somewhere, and the room conditions deteriorate really rapidly because it's taking too long to get those waste products out of your cell. Same thing here. If the cell gets too big, it takes too long to get those waste products to diffuse out of the cell. It takes too much time. The cell conditions start to deteriorate. The cell itself might die. The reverse is true. Let's say you were allowed to get food, and the food was located right down the hall. Well, it wouldn't be a big deal. Just walk down the hall and get your food. But what if it took you 2 or 3 days to get food? That would be uncomfortable, but maybe bearable. What if it took you a month to get the food? Well, now the conditions have become so bad, people will actually be starving. Same thing with the cell. If it's too far, it takes too long for energy materials to get into the cell, then the conditions deteriorate, the cell suffers. So the cell has to be small so that materials can diffuse easily into the cell and the cell can get the energy it needs. If the cell is small, that's easy. If the cell is too large, it gets too hard. Now we can describe the solution that things are in. So here we're going to show 3 different kinds of solutions. hypertonic, hypotonic, isotonic. We're talking about the solutions. So we're thinking about what the mixture of solutes and, in this case, a solvent, water. So we're talking about the water. So let's call it in the beaker here. I've got some very salty water. Here's my cell. Now notice I have 1, 2, 3, 4 molecules of water, and I have lots of red for salt. In my cell, notice that I have lots and lots and lots of molecules of water. Notice that I only have a few molecules of salt. In this case, which direction is the water going to go? Well, I have so much water in my cells compared to the concentration of water in the beaker that the water will leave the cell and the cell will shrivel. Water, water everywhere, and all the pores did shrink. Water, water everywhere, and not a drop to drink. So why is it bad to drink ocean water, like the guy in the rhyme of the Ancient Mariner? Why shouldn't he just drink ocean water? Well, it's so salty that it would actually dehydrate the cells in your body. Water would leave the cells in your body because the concentration of water is so low in the ocean and the concentration of salt is so high. In this case, I have the opposite effect. In a hypotonic solution, I have the opposite effect. So here's my beaker, here's my cell. In this case, I have a whole bunch of water out here. In my cell, I don't have very much water. But in my cell, I do have a lot of salt. So in this case, I have water rushing into the cell, swelling it up, and maybe even causing it to burst. So let's say you go to the emergency room and they want to give you an IV. And in the IV they say, well, you look like a nice, wholesome young person. We're just going to give you pure water. We're just going to put pure water in your IV. Now, does that sound good? I mean, pure water into your body. Well, it might sound good except for this problem. Putting pure water into the bloodstream would cause those blood cells to swell and maybe burst, which would end up killing you. Why do they swell and burst? There's a higher concentration of water in the solution than there is in the cell. Hypotonic. So I think of it like this. Hypotonic, like a big O. Hypo, it swells up to a big O and maybe bursts. Isotonic is just right. Too little, too much, just right. The concentration of water and solvents, solutes, the concentration of water and solutes are identical. So saline solution that you put in your eyes, that's really in the IV, it's identical to what your blood is. And so you have an even exchange without any negative effects. So let me show you a little bit about what that would look like. All right, so hyper and hypotonic solutions. So, for example, I can set up a membrane here. So I'm going to set up a membrane. I can decide here whether or not my membrane happens to be permeable or not. So in this case, I'm going to decide that I have a semi-permeable membrane, where some things are allowed in, but not everything. So in this case, I'm going to allow the water to move, but I'm not going to allow the, let's call it sugar to move. All right? So now the water can pass through, but the sugar cannot pass through. So notice what I've created. Over here, we'll call this side A. Over here, we'll call this side B. Now I have a high concentration of water in side B, a low concentration in side A. If I allow that to be semi-permeable where both can move again, I'm going to notice the water going from here to here. If side A is the cell, then we would call this side B a hypotonic solution, because the cell is in a solution where water is moving into it. So let's make that happen. So we're going to allow both now to move here. So I've got this high concentration of water now moving in, hypotonic. Facilitated diffusion means we need some help getting in and out of the cell. It's still diffusion, no energy required, but we need some help. In the case of water, we can't just diffuse here through the plasma membrane. We have to go through a protein gate or channel. Interpurons is what they're called. So water molecules have to move to these special gates, facilitated diffusion, where it requires the help of a transport protein. Water needs that because oil and water, lipids and water don't mix. So water always flows from a high concentration to low. So take a look here. What's different now in this case? Well, we have something, sodium, and we have some potassium. We have this thing called a sodium and potassium pump. We have our lipid bilayer. We have the inside of the cell and the outside of the cell. But let's take a close look at what's happening. Notice that I have a high concentration of purple on the outside of the cell. That purple is our sodium. Notice that I have a high concentration of gray on the inside of the cell. Now, normally we would think that the purple would be going in and the gray would be coming out. But watch what happens here. Here, instead of the purple coming in and the gray going out, the purple sodium is being pumped out and the gray potassium is being pumped in. Now, notice what it requires for that to happen. Notice that in order for that to happen, this molecule right here called ATP is going to activate that pump. This is called active transport. The transport is going against the concentration gradient, and it requires energy. That didn't happen with our other examples, osmosis and diffusion. But with active transport, we go against the concentration gradient, and it requires energy. Here's some other examples of active transport. Endocytosis means taking things into the cell. So here might be a white blood cell engulfing this bacteria so that the lysosomes can destroy it. Endocytosis. The opposite is exocytosis. Again, requiring lots of energy, going against the concentration gradient. Here might be an endocrine cell releasing some hormones to the bloodstream. Now we can do a little experiment. In our experiment, we want to see what happens when we put our potato cells into different types of water. So the first thing we're going to do is weigh our potato. 10.1 grams. Then we plop it into some normal water. We wait for a while. After we're done, we weigh the potato again. In this case, the potato has picked up some weight, 11.2 grams. And we can find a difference of 1.1. Now, if the potato cells are swelling, that means that I put it into a hypotonic solution. They're swelling like a big O. So now I'm going to take a potato. I'm going to weigh this one, 10.5. And I drop it into some salty water this time. Salty water. So let's see what happens in the salty water. After a while, I weigh it again. In this case, it's gone down in weight, 10.4. I've lost a little bit. Why? Because salty water is a hypertonic solution. Water is leaving the potato, going into my beaker. What about this very salty then? I weigh it, 10.3. I plop it into the very salty water, and I wait. After I get done, I'm going to weigh it again. In this case, it's gone down to 9.3. I've lost a whole gram. Why? Because it's very salty. Water's leaving the potato cells, entering the beaker, making my potato weigh less. So if I were to drink this, I would expect the same thing to happen to my body cells. Water would leave my body cells, and I would be more dehydrated. So if you put a potato in very salty water, it will gain mass, lose mass, stay the same or grow. In this case, lose mass. Maintaining proper balances between solvents and solutes is a big job for the cell, and it's difficult. And the cell works hard, sometimes expending energy to do those kind of things, like active transport, the sodium-potassium pump, working hard to get that sodium-potassium against the concentration gradient. Your nerve cells do that really well. This is a major part of the cell's life, using that plasma membrane, trying to maintain proper balances so that the cell can maintain a healthy environment.